the Wagner Lab's custom SPRI DNA extraction instructional video. In this video, we will be thoroughly following the steps from this protocol, which can be found in Benchling. My name is Savannah Parker, I'm a lab tech in the Wagner Lab, and I will be giving you some of the tips and tricks I've learned from doing these extractions over and over again. To begin, let's make sure we have all the materials and reagents we'll need for the entire extraction. Recipes for each reagent listed here can be found in a separate Benchling entry titled Custom SPRI DNA X Reagents. Be sure to calculate how much reagent you'll need ahead of time. This reduces any chance of inconsistencies throughout the project and saves you a lot of time. Let's get started by preparing our bead beading plates. We'll start by filling this 0.45 milliliter plate with yellow zirconia beads. And we'll end up using these plates for homogenization to break up the sample and release DNA. Once our 0.45 ml plate is full, we'll put this empty 2 milliliter plate on top of the bead plate with the openings of the wells facing each other. We'll flip this over, and it's always helpful to do this over a bin to prevent any spillage. Now we'll add 2 soda lime glass beads to each of the wells. We'll use this nifty sifter to do our best to get 2 glass balls into each well. The sifter isn't perfect, but using tweezers helps a lot. For root extractions, we'll be using these cluster tubes and putting 2 steel balls into each of the tubes to break up the sample and release DNA. I'm starting here by labeling each side of the cluster tubes from A1 to A12 and H1 to H12. We'll use our nifty sifter once again to get the 2 steel balls into each of the tubes. Now that our plates are full, they can go into the autoclave for sterilization. These plates require a 30-minute gravity cycle. Now that the plates have been sterilized, we can mark them up and label them with the designated plate IDs. We color the bottom left corner of the plate to keep orientation consistent throughout the entire extraction. Here, I'll show you how we add our soil samples to the bead plate. For our first well, we'll measure 250 milligrams of soil. Once we have that, we can eyeball the rest of the wells based off of our first measured well. Here, I'm measuring the size of the well to cut out a hole in the tape. This prevents soil from falling into other wells during the transfer process. It is always a good idea to add a positive and negative control to the last 2 wells of the plate. Positive controls can be found in the -20 freezer. Also, be sure to note which sample goes into each well in your lab notebook. Now, skipping to step 14, we'll do the same thing for our root and rhizosphere plates. We want to take about 2 centimeters from the newest point of the root tip. Be sure to wipe down your work surface and sterilize your forceps and scissors in between each of your samples. Now that our samples are loaded, we can add 800 microliters of lysis buffer to each well using our multi-channel pipette. Lysis buffer helps us isolate DNA by breaking down the lipid bilayer of cell walls and membranes, which then releases the DNA into the solution. This is another good stopping point, so if it works in your workflow, you can store this plate in the -20 freezer until you're ready to continue. Once our samples have thawed, we can add 10 microliters of 20% SDS into each well. Our SDS solution breaks up cell membranes, similar to lysis buffer. It also works for protein denaturing and will help release DNA during the homogenization process. This is our homogenizer. We'll insert our plate and tightly secure it by twisting the knob. Then, we'll close the lid. We're going to do 2 cycles of 1200 RPM for 5 minutes. To remove the plate, push back the metal lid lock and unscrew the knob. Make sure you wait 1 minute between each cycle to let the homogenizer cool down before continuing. Homogenization helps physically break down the sample, while our lysis buffer and SDS solutions help chemically break down the sample to release DNA. Our next step is a 90-minute incubation at 55 degrees Celsius. Since this is the step that most often causes contamination, I like to add an extra layer of protection. I do this by securing a plastic lid to the top of the plate with rubber bands. This prevents the samples from leaking into the other wells. This incubation step promotes enzyme and proteinase K activity. This will help release DNA while preventing degradation due to the high temperature. So after the 90 minutes are done, we'll take our plate out and prepare it for the centrifuge. We'll take off the rubber bands, the lid, and we'll reseal the mat by pressing down on it. Now we'll centrifuge at 3600 RPM for 7 minutes and 20 seconds. While our plate is in the centrifuge, we can fill a new 1 milliliter plate with 130 microliters of 5 molar potassium acetate. Be sure to label the plate before you start filling it. Once the centrifuge cycle is complete, we can transfer the supernatant to our new plate with the potassium acetate. Make sure your plates are in proper orientation by aligning the mark you made at the bottom left corner. We'll transfer 400 microliters of the supernatant to the new plate, changing pipette tips in between each column. Do your best to avoid the pellet by keeping the tips of the pipette against the wall of the well. Also, hold on to that plate with the roots. We'll use that later. Now we'll reseal the mat onto the plate and prepare it for vortexing. We'll vortex for 5 minutes at 100 RPM. After vortexing, the plate needs to go into the freezer for at least 30 minutes or overnight. Going back to step 8, we'll take the root plate we kept from the last step and transfer each root into the cluster tubes. Be sure to sterilize your forceps in between each sample. Also, be sure that your plates are aligned in the proper orientation. Once our root plate is full, we'll place aerosol film on top of the tubes. This film allows carbon dioxide exchange and air filtration during the freeze-drying process. We'll come back to this plate later, but for now, store it in the -80 freezer. Now on step 25, take out your samples and allow them to thaw before putting them in the centrifuge for another 7 minutes and 20 seconds at 3600 RPM. While in the centrifuge, we'll take out the syrup here and allow it to come to room temperature. We'll mix it around until all of the beads at the bottom have mixed into the rest of the solution. Syrup here is light sensitive, so rewrap the bottle in foil before moving on. We'll also need to warm up our TE buffer to about 40 degrees Celsius. With our syrup here at room temperature, we'll mark up a new 1 ml plate and add 600 microliters of syrup here into each of the wells. The speed beads in the syrup here solution are made from iron oxide, which facilitates the magnetic separation of DNA from the rest of the solution. Once the centrifuge cycle is complete, there should be a pellet of potassium acetate at the bottom of the wells. We can now transfer 400 microliters of the supernatant to our prepared syrup here plate. Once the transfer is finished, place the plate on the vortexer for 5 minutes at 100 RPM. Incubate at room temperature for 2 minutes and pop that plate back into the centrifuge for another cycle. After the centrifuge, place the plate on top of the magnet rack for 5 minutes. The magnets will hold the beads containing the DNA to the bottom of the wells. This will allow us to discard the supernatant. Discard by tightly squeezing the plate and the rack together and dumping the supernatant into the sink. Trust the process, the beads will stay at the bottom. Now we'll wash the DNA with 80% ethanol. Add 900 microliters of ethanol into each of the wells. You can reuse the same pipette tips as long as they don't touch any of the beads at the bottom of the plate. Once you added all your ethanol, allow the plate to incubate for 1 minute before you dump it into the sink. We'll do this for a total of 2 ethanol washes. After the second wash, tap the plate onto a stack of Kim wipes. Then, allow the plate to air dry for 3 minutes. Now we'll rehydrate the DNA by adding 75 microliters of TE buffer into each of the wells. You can reuse the same pipette tips as long as you don't touch the beads at the bottom of the plate. Now we'll reseal the plate and vortex for 5 minutes at 100 RPM to mix up the beads into the TE buffer. While vortexing, label a fresh 0.45 ml plate with gDNA following your plate ID. The centrifuge plate will go on top of the magnet rack for 2 minutes to isolate the beads. Then, we'll transfer 75 microliters of the supernatant into the new gDNA plate, avoiding the magnet beads at the bottom. Now we have all the genomic information for each of our samples wrapped up in one single plate. We can store this plate in the -20 freezer. You can keep this here until you're ready for further sequencing. But wait, what about our root samples? After 24 hours in the -80 freezer, they're ready for freeze drying. This is our lyophilizer. We'll start using this by disconnecting the drain hose. Then turn on the collector and allow the temperature to drop to -50 degrees Celsius. Hit the switch on the left side of the tray dryer to turn it on. After 20 to 30 minutes, the temperature should drop to -50. Now we can place our plate into the lyophilizer. We'll place a temperature sensor into one of the tubes to keep track of the temperature. We can track this on the program screen. Now, turn the tray dryer handle to the locked position. Enclose the vacuum release valve on the side of the tray dryer. After 25 minutes, check that the pressure has dropped to less than 1 millibar, and then start the 48-hour program for freeze drying. And once complete, turn off both the collector and the vacuum and reattach the drain hose. Turn the tray dryer to the open position and remove your samples. They are now ready for homogenization. Here we'll remove the aerosol film and add cluster tubes to each of the tubes. This will keep the samples within the cluster tubes throughout the homogenization process. Now we'll start our homogenization process by doing a 1-minute cycle at 1200 RPM. We'll continue to homogenize the samples until they turn into a powder-like form, something like this. Once the roots have been powdered, remove all of the strip caps. Once all the strip caps are removed, add 850 microliters of lysis buffer to each well by pipette mixing up and down 3 times. It's helpful to cut the tips of the pipette to allow all the root material to go through and to prevent clogging. After mixing, transfer the solution into a fresh, labeled bead plate. This plate could be stored in the -20 freezer, or you can continue the extraction from step 18. Extracted, it's time to clean up. Collect the steel balls from the cluster tubes and keep them for reuse. They need to be autoclaved before being used again, so make sure you label whatever storage container you're using.